Welcome everyone to the MTL seminar this week. So today we have Dr. Zhang, Professor Zhang from Purdue University. Dr. Zhang is an assistant professor in the Department of Chemistry at Purdue University. He earned his PhD in 2014, not that long ago, from University of Michigan, specializing in surface nonlinear optical spectroscopy. From 2014 to 2020, his postdoctoral work at Purdue University, Boston University, and University of Illinois at Urbana-Champaign, where we met, focused on nonlinear optical spectroscopy and imaging for biomedical research. He began his independent research career at Purdue University in 2020 within the analytical division of the chemistry department. Oh, so you guys have a division within the department. Nice. Okay, so very clear. Dr. Zhang's current research focuses on developing novel optical imaging and opto control technologies for biological applications. He has published over 50 peer-reviewed journal articles. His lab has pioneered the RPOC technology, which we will hear about today, which enables precision optical manipulation of chemical processes in live biological samples. Fascinating. So his research is funded by NSF, NIH, Merck & Co., Purdue Trask Foundation, and the Shell Water Foundation, I'm probably butchering the names, and the Center of Bioanalytical Metrology. Notably, he received the Mira Award with multi-million-dollar support from NIH. Additionally, Dr. Zhang is the founder of Photokinesis, a company dedicated to the commercialization of advanced optical control technology. And Dr. Zhang, please take it away.
Thank you very much, Zhixian. It's my well, thank you so much for your introduction and then for your invitation as well, so that I get a chance to share my research with the colleagues, experts in MIT. And I overlapped with Zhixian at the Stephen Ballpark Lab for I think about 2 years. So it was a great time when we discussed science, right? And we work in the same room, I think. Oh, not the same room, but I see you. Zhixian worked very hard and published a lot of great papers there, and it was a great time. And I also worked at BU for 1 year before I joined the Steve Ballpark Lab. So I'm familiar with this area. I haven't been to MIT before, but I was in the other side of the river a lot. So it's great to share with you some of the research that we have done at Purdue. So since Zhixian is in the engineering department, so I put the engineering first. So although I'm in chemistry, the reason I put it here because we're in analytical chemistry, so we did a lot of engineering in chemistry. So basically, you can consider we're the engineering section of the chemistry department. So. Well, the talk is controlling and imaging and controlling chemical reactions. I think the imaging part overlaps a lot with what you're doing here. And then the controlling part is slightly different, and that's also another direction we started. So if you look at the imaging part, we did a lot of chemical imaging, including the coherent Ramans or spontaneous Raman spectroscopy, fluorescence imaging. We did a lot of fluorescence after I started my own lab. And then along that direction, so we put half of the energy on developing new tools, like a new system to improve the imaging technology, to improve the performance. And another half to delve into the biological questions, like how cells is changing in stress conditions, like in the low oxygen condition, in all different kind of toxins. And we look into the lipid metabolism, cytoskeleton, etc. So on top of that, we developed a new direction, which we're pretty proud of. So that is controlling chemical processes. This is more of actively engaging in the cell biology or functions of the cells. You can change or control the cell, how the cell behaves using the technology we call RPOC. Well, this is the development of this technology, now pushing it towards the commercialization, and we are also exploring a lot of biological applications, and we'll talk about today. So I highlighted some of the red items here as just to show as examples of each category. So and then the black ones are ongoing projects, and probably don't have time to cover. So. All right, so I want to move on to, I'll start from this side to that side. I'll do the imaging first. I start from fluorescence. I think you're probably familiar with fluorescence, right? So fluorescence is a very highly sensitive and also very high resolution, and you can get very good chemical specificity, especially for looking at proteins, because you have many good ways of tagging things. So one way for dead cells, you can put an antibody there to label almost any kind of protein, and then put another secondary antibody to label the primary antibody and see the distribution of that protein. So for live cells, you can do a genetic transfection, so to link a fluorescent protein to any proteins of interest. So here, what you see is engineer this EGFP tagged something called EB3, end-binding protein. So what you see, this is basically the dynamics of that protein, which binds to the plus end of the microtubules. So what you see here is just the microtubule polymerization in real time. So you can actually use this. You'll see this again. So aside from that, we do a lot of Raman. So the Raman spectroscopy is a label-free technique. So and Zhixian did a lot of label-free. So people like label-free because you don't have to put the labels in it. So the reason we like Raman is you don't have to put dyes, and then you can use basically what Raman does is you put lasers interact with the sample, and the laser interact with the sample and exchange energy with the vibrational modes of molecules. And then they can shift the input photons to different frequencies. Once you measure the frequency, you can measure the chemical composition in the sample. So this conventional Raman is pretty low in efficiency. So then in order to improve this Raman efficiency to make imaging faster, people developed this coherent Raman. So my postdoc advisor and his advisor developed this CARS and SRS. So what I mean, a little bit of chemistry here. So basically you have two lasers, one called pump, the other is called Stokes, and sent together to the sample. And then the beating frequency of these two synchronize the molecule vibrations. So the molecular vibrations are synchronized, so they generate a coherent light that is very strong compared to the spontaneous one. So it's called coherent anti-Stokes Raman. And also these two lasers actually make the quantum efficiency of energy coupling from lasers to the molecular system more efficient. So they created more vibrations at the same time, change this, make the annihilate more pump and create more Stokes photons. Basically, you can consider that this new technique improves the signal level to 1,000,000 times or even higher. So you can image them as fast as fluorescence. So we used a lot of nonlinear optical modalities, like when Zhixian was using the SLAM, right? You integrate the multiple imaging modalities. We do that too. We can integrate all these into a single platform. So you have the coherent Raman, stimulated Raman, two-photon fluorescence, second harmonic generation, etc. So you can detect different molecules using different modalities, depends on which one you prefer, right? So that's some of them are very sensitive to non-central symmetric materials. Some of them are good for drug molecules. So we integrate them into a single platform. It looks very complicated, but this is the first system we built, and we have all this contrast that can be generated. I'll give you a few examples here. This is a lipid droplets moving in the neutral lipids in the live cells, so they're very dynamic. They're moving around. And this is the neutral lipids in liver tissue, in the NADH, in the same field of view, and the mouse tail tendon, etc. So these processes are nonlinear. So what does it mean? It means that if you put in the light, like you increase by a factor of 2, your signal changed nonlinearly. So either a factor of 4 or even higher order. So what you see here, you see the input intensity squared proportional to your output signal, or even the product or the higher order. So this gives us a way to improve the sensitivity. So because you can see that if I increase the peak power, so I will increase the output intensity, right? So the way is that how to improve the peak power. So let's see how usually people do this imaging. I have an image of 400 by 400 pixels, 10 microseconds per pixel. It's a typical number. And I use 80 megahertz laser. How many pulses do I have for each pixel? Or you can do a simple calculation, so you get 800 pulses. So this is not the best way of doing it, because you don't need 800 pulses for each pixel, right? You only need a few. So, so the best way, or the more efficient way, is instead of having this many pulses for one pixel, you can actually remove these pulses and put that energy on top of each other, right? So then you can actually get a much higher signal. That's because although you reduce the number of pulses, you only have fewer pulses, but each one has a higher peak power. So, and your dependence of that signal is nonlinear, but your dependence with the number of pulses is linear. So that's why you can get a higher signal level in general, right? So we built this, based on this concept, we built this pulse picking nonlinear optical imaging technology. So we use this AOM as a pulse picker. So basically what it does is when you apply a voltage, it sends an acoustic wave and deflect the beam to a new direction. So then you can basically let this pump and probe pulses to be simultaneously sent to a new direction and pick them nicely, so you can reduce the duty cycle, and then you can let fewer pulses go to your sample, and then do imaging. Then you can increase the power for each pulse, and then to have the same average power, but the peak power is much higher. So this is a way to better use the system because we have a lot of power for the laser source. You can't send all that power to the sample. Usually people dump like 90% of your power at the beginning. So, and a better way is to dump it at the AOM. So instead of dumping it at the average power, you dump the number of pulses and use the full power at the beginning. So you can improve the sensitivity very significantly for cars, for two-photon fluorescence. Cars is about 1,000 times. So, and then for two-photon fluorescence, it's about 20 times. So if you look at the image contrast, it's very significant, right? If you compare no pulse picking, almost you see nothing. And then if you do the pulse picking, just reduce the duty cycle. It's basically a very simple device. You put it into the system, and put a function generator, that's it. So then you can improve the contrast very significantly. And you can also do the spectroscopy imaging because Raman, we care about spectral information. We can take a spectrum, hyperspectral image, and you can see what are these molecules in there, and by taking the spectrum for each pixel. So when you do Raman or coherent Raman, people care about spectral resolution. So usually we use picosecond laser because picosecond laser compared to femtosecond is very wide in time domain, very narrow in the frequency domain, right? But if you work with people in the nonlinear optics for two-photon fluorescence, people say you should use a femtosecond laser, right? Because the peak power is higher. It's actually partially true because peak power higher is needed, but it doesn't have to use femtosecond laser because picosecond laser, if you use the same pulse picking method, it's even higher than the peak power is even higher than the femtosecond laser. So it's the same thing. So then, before I move on, I want to say a few words here. You can perfectly integrate the nonlinear optical image of Raman and the multiphoton fluorescence by just simply using picosecond laser. I can show, go back, and so you can see that I just use 1.5 milliwatt, 3 picosecond of laser. I can just single frame, you can get a very nice two-photon image. So the peak power is higher, even higher than femtosecond laser. Anyway, so we also developed some tools to expand the spectral coverage of coherent Raman. Usually people cannot measure wave numbers below 400. So I didn't have time to put more details here, but I just want to show you that we also make some technological improvements to cover the spectral range below 400, and the lowest we can reach is about 83 wave numbers. So we can image a lot of chlorinated compound like this DDT and some other drugs that has chlorine in it, and also the inorganic materials like the sulfur, like some calcium fluoride or something. Usually you don't see any signal in the Raman. Yes?
A question. So if you use lower rep rate laser to increase the peak power while keeping the average power the same, how do you do the modulation in SRS to match that 700K kilohertz?
Oh, great question. You mean you're talking about the previous one, right? So here, this is basically it's equivalent of changing the repetition rate. It's the same thing. It's just you can go to any repetition rate you want. It doesn't matter. You don't have to change your laser, right? So you basically just use the full power. So then you can actually, this one doesn't really matter. You can change to anything. And then what matters is the duty cycle. It's just, if you have 80 megahertz laser, then if your duty cycle is 2, that means equivalently you divide it by a factor of 50, right? So basically that becomes 1.4 megahertz. So it's the same thing. But for SRS, it doesn't work very well because this modulation could couple into the SRS detection. So we so far haven't get the SRS work. But for coherent Raman and two-photon fluorescence, it works perfectly. Yeah. Thanks for the question. Yeah, and I also want to talk about some phototoxicity because we do biological imaging, we care about phototoxicity, right? So, and how the phototoxicity is generated. So there are two major pathways. One of them is linear absorption. Basically, you just heat the sample too much. You have too much average power. The thing just heat up and then become, and then create bubbles, water bubbles. You just boil it. Another way is you generate too much peak power. So the peak power induces multi-photon absorption. So basically, you just ionize the molecule. The electron fly out after this. So you can't have either case. You have to use something in between, right? So there's a nice window there. So to do that, you want to keep it safe. So, and this is the average power, and this is the pulse peak power map, right? Usually, for any imaging system, you have a limit of detection. So the combination of this has to go above it, otherwise you don't see anything, right? And then you have a damage threshold. And that damage threshold means if you go above it, the sample will be damaged. It's useless. So you have to use this range, right? This nice range. So if you have a fixed repetition rate laser, well, it's always tied together. So you increase the average power, your peak power also go up, right? So you can only access this range. So if you go lower repetition rate, you can probably go here and here. So if you use the pulse picking, you can actually access the entire thing. It just use the same laser. You don't have to worry about where this line is going to crossing it. So basically, you're separately tuning the peak power and average power. So it's a very simple device. And if you ask me how do you quantify the phototoxicity, there are many ways there, but those are not good enough. For example, you can see the cell is shrinking, the cell is being membrane blabbing or something. Those are too late. In those stages, the cell is already dead. So we need more accurate way or more sensitive way of measuring it. So it turned out that this small molecule concentration or small molecule dynamics is very sensitive. So one way is you can actually measure the ATP concentration. But if you put the ATP dye in it, well, you already perturb the cells. So you can do it in a label-free manner, basically just measure this lipid droplets dynamic on the sample. Because the droplets is powered by the ATP. If the ATP is gone, then this thing's movement is greatly reduced. So you can quantify that, drawing the histogram and measuring this lipid droplets dynamics, and you can quantify in which case, in which power range, this laser is safe for live cell imaging. We usually consider that less than 24 milliwatts on the sample and less than 36 watt peak power is using the IR laser is safe for cancer cells. For different cells, this is very different. But this is still not sensitive enough. Recently, we found that the one you saw earlier, this guy, is way more sensitive than the previous one. So this is a fluorescent protein that is basically binds to the plus end of the microtubule. And this process is powered by the GTP. So it's another energy currency. So if the GTP is gone, this thing will immediately disappear. So this is the best way, I think, to quantify the phototoxicity. Because if you perturb the cell a little bit with some fluorescent dye, with lasers, this thing changes very, very fast. You can even see the changes in about 1 second sometimes. So they flashed away. So we use this to quantify the commonly used fluorescent dyes and then to see how how toxic they are. And you can quantify that and generate a map to quantify that. And then also you can correlate it with the excitation laser wavelengths, the specific organelle, which organelle is more perturbed and is more toxic to the cells. Those are recently we just achieved. And we also quantify that in the low oxygen conditions. So we developed this incubation chamber so you can image a low oxygen condition. We cut the oxygen supply. So here's a little bit of chemistry here. So if you think about if you cut the oxygen, if you choke the cells, what do you expect? The phototoxicity going to go up or go down, right? So most people will think it's go down, right? Because the reason the phototoxicity happen is your photon energy get absorbed by some kind of photosensitizers, either FAD or the dye you put in, and that electron or energy transferred to oxygen create a singulate oxygen or other like H2O2 or something, and then eventually damage the cell. So if you don't have oxygen, the whole thing should reduce. That's true most of the cases, but in some special cases, this is the opposite. So when we target the mitochondria, if you label this mitochondria with the mitochondria dye or put some drugs on targeting mitochondria, this is the opposite. In the low oxygen condition, the cell died even more than in the normal oxygen condition. So this is some hints for us that in the future for photodynamic therapy, which usually doesn't work very well for low oxygen condition, we should have a chance of targeting mitochondria so that you can kill the cancer cells more efficiently in the low oxygen conditions. All right, so I want to continue to talk about the low oxygen conditions for a few slides. One of them is, the reason why we studied low oxygen conditions is because in the solid tumor, because of the disruption of the blood vessels, usually the environment of a cancer cell, and is in a low oxygen condition, right? So we want to study how lipid droplets, or in general, how lipid metabolism is changing in that condition, and how cytoskeleton is changing in that condition. So I've just used 3 slides for each case. So to describe it, the first one is we found that the lipid droplets, the one you saw, those organelles, accumulate on the ER from many of these cancer cells in the hypoxic condition. So this is a new discovery. So if you ask me what are the lipid droplets, I can tell you those are organelles are found in most of the mammalian cells, and they are enriched in neutral lipids. If the cell eat too much fat, for example, and they cannot tolerate it, and they're toxic, so they pack them into neutral lipids. So they store in those droplets, and they can still metabolize them, use them. So you can see them very well by just tuning the Raman to this CH2 transitions. You see all these lipid droplets nicely. And then we can quantify them, we can analyze them very nicely. For example, we did this time-lapse coherent Raman imaging. You can see how these droplets are, lipid droplets are, at the beginning, there's everywhere. It's both on ER and outside ER. And after you expose it to low oxygen condition for about an hour, all those droplets release from ER and then get into the cytosol. And after about 24 hours, so this lipid droplets reaccumulate on the ER, and they cannot shed it off anymore. So that's can be quantified, and then this is another example showing you that how this they have very different dynamics, because it's on the ER they can't move it, they can't move much. On the outside they're moving on the microtubules. So anyway, so we developed this IB-CAR system, so and also the all automatically controlled and remote control system, because otherwise the student have to stay in the lab for 24 hours. So we designed this, the student can do it at home and in the swimming pool or something. So they just need to press the button. Once you put a cell on it, you don't have to come to the lab anymore. So that's a bad thing, because the students really stopped showing up in the lab, and I couldn't find them to talk to about the results. Anyway, so this is another example of, we also studied a lot of biochemical, we studied the biochemical mechanisms, like how this lipid droplets are formed, how which nutrition they prefer. So long story short, we found that these pancreatic cancer cells, they prefer more lipid uptake. They directly take up from the from the medium. And then if you look into the LD synthesis from glucose, it also happens there. We have to use like a post-chase analysis here. Basically, you have to feed the cell with a deuterated or isotope labeled lipid, and then see how that deuterated glucose converts to deuterated fatty acid. So those can be done, but this is less preferred for the cell compared to the first one. And we can also did a lot of, we can also analyze the lipid droplet content change, like how, if you look into one droplet here, and how this content, such as the cholesterol ester content, the lipid saturation level changes in this, in different conditions, we can analyze all these specific composition changes. So aside from the lipids, we also look into the cytoskeletons. So for example, this is the time lapse over 48 hours. You can see how this cancer cells start to grow protrusions like this very significantly, right? These what you see here are the signals from microtubules. So they are microtubule polymerization process. They're protruding out, and then if you do some biochemical assays, you can find that if you control the nutrition level, you see that these ones are triggered by the glucose. If you cut the glucose, nothing is going to happen. There's no protrusion. So they grow because they have a stronger glucose metabolism. And if you ask me why the cells are doing this and what are they, what's after, so the cells are actually migrating faster using these protrusions. If you see the hypoxic condition, normoxic condition, this one closes the gap even faster than the normoxic condition. So that's because these protrusions are helping the cells to migrate to a new location. And then this is usually known as the epithelial mesenchymal transition. So that's the process that helps the cells to become more invasive in the hypoxic condition. So they metabolize that, they change their metabolism and make these significant phenotypic changes. So and we also did a lot of study on what causes this and what specific organelles is related to it, and I just want to mention one quick thing is that the mitochondria function in that protrusion is related to it. Basically, we saw the mitochondria migrate into those locations, and we also find a specific enzyme that is related to that converting ATP to GTP process, and that's because what powers the microtubule polymerization is basically the GTP. So anyway, but here there's one thing we want to prove, right? We want to prove the mitochondria function in the protrusions is tied to the microtubule polymerization here. So there's one easy way of doing this is basically just inhibit the mitochondria functions here and see how this protrusions is changing, right? But there's no way to do it because if you inhibit the mitochondria function, you inhibit the entire thing, right? You can't prevent them inhibiting it here because you treat the cell with drug and drug diffuses everywhere. So this actually leads to the next topic, so which is the question like how to precisely control the organelle functions and drug activities in cells. So we wanted to do it precisely. So we basically, the long story short is we wanted to, instead of have the drug to be active everywhere, we wanted to be only active at the location you want it to be active. So conventionally, if you have worked with cells, you know this is how you do it. I culture the cells, I put some chemicals in it, and I measure it. And then basically, the cell is going everywhere, and it might interact with the target. It might interact with somewhere else, that some other molecules you do not want it to interact. But you have no control of it because it's just the nature of diffusion, right? So the topic or the concept that we introduced here is called RPOC. This is the real-time precision optical control that we developed. So this is different from imaging, but it's built upon imaging. So what we do is we scan the laser on the sample. If you have ever worked with nonlinear optics or confocal microscopes, you know the laser scanning, basically you scan it quickly, you generate the image. So the laser is scanning on the sample, the red one generates signals at some places where your molecule is located. If you collect the signal, that's just the first part of the talk, it's chemical imaging, right? So we take another step forward is instead of just measuring this, once you see something, we quickly turn on another laser, which is we call the action laser. So this laser will do something. This laser will trigger the chemical processes that you want. So that's the action laser, which is does the job for you. Okay, so that's the second process. And the beauty of this is a very fast feedback control system. There's no, you don't have to take an image and then say, I want to do here, here, here. Basically, while you're doing imaging, this control is done. So basically, the feedback control is done within hundreds of nanoseconds. It's very fast. So that is shorter than the pixel dwell time. So you don't need any prior knowledge of the sample. So there are three components for this RPOC. The first one is, of course, we want to ask where we want to control it. Right? The second one is how to control it. And the third one is how the cell responds to it. These are the three basic questions there. And the first one is basically done with the excitation laser. Just like our imaging system, we have the excitation laser. The second one is done with the action laser. It's the laser we usually use to trigger the chemical process. And the third one is a readout laser that basically give you a readout and see how the cell is responding to it. But usually some of them are interchangeable. And the one where the chemical reaction is going to be controlled is called active pixels. So that's where the chemical is going to be controlled. So these three things are simultaneously performed at each pixel. So which means that you don't have to come back and then do it again after imaging the sample. So each component has a list of things you can do. Right? For example, you can use fluorescence, Raman, IR. Anything that generates a contrast of optical signal, you can use it to identify your target. And then the next one is you can use many lasers to do the chemical control. You can do the blue laser. Blue laser does generate the reactive oxygen species. Right? It can activate a drug. It can do a photo uncaging. It can also do photo bleaching, and blue light can also do the same, but just for different molecules. And you can use near infrared to heat some samples. Specifically, you can use plasma, a femtosecond laser to generate plasma for using a different mechanism to control it. So for the last part of the readout, this is even more informative. So they have short-term and long-term responses. Short-term including the protein dynamics, how this microtubule polymerization is changing, how the calcium signal is changing, and then organelle function is perturbed. And then long-term including you can watch the cell division, cell migration, viability changes. So this is not easy. This is pretty difficult because we have to only work with live cells. It doesn't work if you fix the cell. It has no response if you treat it. So only live cells, and we have to keep live cells very happily at the location for 72 hours to monitor the long-term study. So how to do that? Well, you have to check all these boxes. So we already checked the first one, chemical imaging. That's the first part. And I will talk about the closed-loop feedback action laser, which is the one that is going to do the job. And stable microenvironment, we solve that problem. And then we also evaluate the phototoxicity. So we know that the imaging laser is not creating phototoxicity, but only the action laser is doing it. So I'm not going to talk about this in this order. I will talk about it in a chronological order. So the first time we developed this technology is based on the SRS system. Because at that time, we only have a stimulated Raman system in the lab. I did this as a postdoc, so that's the only thing I know how to do it. And we don't even have an action laser. And we don't even call it action laser at that time. So we have to convert the IR laser to visible and blue and the green lasers by frequency doubling, and then couple them in. And AOM is basically a shutter. And then once you do the imaging and then the signal sending to the comparator circuit, make a decision and send the AOM to open or shut. So that's how it works. So this one is a comparator circuit, the first version of it. So basically what it does is your optical signal comes in, compare it with some threshold, and if it go above it, you send it 1 out, otherwise you send 0. And it's just do it in nanoseconds response time, so it's very fast. So you can actually shine, the first thing we tested is this, how the system works. It's basically shine any laser, shine the action laser to any chemical species that we can do, right? If you identify a chemical species, then you want to shine laser only to that location and to visualize the active pixels. And you can do it for Raman. At that time, we only have the Raman example. So if you look at this, this is a movie showing you if you tune it from this wave number to this wave number, it's all in real time. You don't have to come back and take an image. Once you go to that peak, and the active pixels is already automatically assigned to it. And then which means that you can apply this to a highly dynamic system. If this thing is moving a lot, it doesn't matter. As long as you see this in this image, you can actually control that molecule or organelle specifically only for that chemical. So we can also do a lot of calculations. Like, for example, I have 2 discriminators. Like one tells you, let's treat chemical A. And the second one tells you, let's treat chemical B. And if you use the N function, it will treat chemical A associated with chemical B, which is like the things like overlapped or something. So it's very flexible. So the first example we did is this chemical, which probably nobody knows what is it. It's called CMTE. It's just a chemical that has 2 colors. So if you use blue light, you can switch it on, and the green light, you switch it off. So then what we can do is we can feed this chemical with cells. The cells take them up, and they accumulate. And you can set a threshold to let some of the aggregations to be above the threshold, some of them below the threshold. And then what is above the threshold, it will turn the laser on only at that location and switch it to the other, turn it off, basically. And then otherwise, it will keep it untouched. So although the distance between them is only like 2 micrometers, so that the spatial precision of this system is about 300 nanometers, so it's very precise. So we can also use the ER signal to say, let's keep this molecule in the off state outside of ER and on state inside the ER. You just need 1 scan, and then all these molecules are being converted to different states. So that's how the precisely control of this molecule, which is not very useful, but they demonstrate how the system works, okay? And when we talked to biologists, they said it would be great if you have fluorescence for this, because they don't know what is SRS, Raman, unfortunately. So then we completely redesigned the system. We disassemble it. We build a new system for confocal fluorescence. We also have the 3 components there. And we have the action lasers as this too. If you look at it, it looks like a confocal microscope, right? 4 lasers combined and then scanning 2 confocal. But it's actually an optical control system. You have the action laser here with the AOMs and comparator circuit to make the decision. And this system is, I will not go through the details of this workflow, but that's the graphic I showed in my flyer. But this is basically pure engineering here. So basically you are making the decision in real time for any color and using a chemical composition when they detect it. So the system is very robust, and we don't have to use it under and turn off the light. You can use it under room light. It's just beautiful. So that's why I turn off the light and take a picture of it. So the cost is very low compared to the first version. It's only 10% of the laser, 30% of the entire system compared to the first system we built. So then you ask me, what can you do with it? So, well, the first thing, the easiest thing is you can shine different color of light to different locations, right? But let's say we shine blue light to different organelles. So we know the blue light is bad, sometimes bad for our health. If you're exposed to too long, they generate ROS. But we don't know ROS, which organelle is more perturbed by it, and which one is more toxic, right? You can shine the blue light to nucleus, to mitochondria, to whatever you choose. You just need to find the signal of it. And then you can study how this, how that blue light interaction is affecting the cell function. So what it does is blue light is absorbed by the FAD. And FAD work as a very weak photosensitizer, and they generate a ROS in this location. And usually this ROS is get absorbed quickly by surrounding molecules, but the H2O2 can diffuse out because it's less reactive. So they can photo bleach or oxidize all these fluorescent proteins. And that's what you see the responses here. You can quantify that by just comparing the results. So you can also turn on and off a drug. So that's something we are interested, right? You want to make the drug active only in the location and somewhere that's not active. So this is a drug we synthesized with the name of PST1. So this drug is basically it's active when you use the blue light to shine to it, and it will be inactive when you turn it back with a green light. So usually you put the drug into the system, you can make the cell. You can let the drug to be active for the entire cell. So what we do is we can actually make the drug to be only active in the subcellular locations, like only in the ER, only in the location that you want it to be active. And basically outside of that place, you will still see the microtubule polymerization. And once you turn them on using blue light only in the ER, for example, that microtubule polymerization will be gone. So you might ask me, what if the drug, after you activate it and it diffuse out? So right, they can eventually inhibit everything here because it diffuse. So yeah, it is happening. It's just if you don't do anything, it's gonna eventually everything gonna be inactivated. So what we do is we shine a green light outside and shine a blue light only in these locations. You can actually do them simultaneously. So once the drug diffuse out, you can convert it back to the inactive form. So that's how you can prevent that diffusion effect to happen. So recently we developed another more advanced technique. So this one is based on as we call it, the human-machine interaction system. So basically we synchronize the laser scanning with the image and with the AOM and with the detector. So what you can do now, you just use a pen on the screen, you just draw the area and select that area, and the laser will just go there. And then you can select green for this, blue for this, or anything, and adjust the threshold and change the active pixels and how much you want to interact with that area. So you can use it, combine it with a comparative circuit. So and these are the workflows of how, basically what it does is you convert an image. Let's say we take an image first, and then we convert the image to threshold it, and then convert that to a time series of TTL signals. And you can, once the laser scans to that same location, they will turn the laser back on. So that's how this system works. So I will not go through the details of this. Is a more advanced comparative circuit. So basically, it's part of the brain of the system. So you're making the decisions using this device. So it's basically the fundamental thing is you can compare your signal with the specific threshold you define, and then make the decision in real time. But I do want to show you that you can do a lot of fancy things. For example, you can draw any area by yourself, and then change this threshold, and now you can determine the active pixels and then photo bleach them. So photo bleaching itself is not very interesting biologically because it doesn't do much. But what I want to show here is you can do it precisely. This is basically showing you how laser is precisely doing some work that you can visualize it. Okay, you can put a Purdue mask and photo bleach it. These are 1 micrometer beads, by the way, so they're very precise. You can photo bleach it down to about 300 nanometer precision. That means that our precision is about 300 nanometers. So you can put anything in there. You can if you put a photo in the system, my student's face, you photo bleach himself on the 1 micrometer beads. You can also bleach him on the nucleus. if you want. So the beauty of it is you can control, you can create multiple treatment conditions simultaneously now. So let's say I want to treat this guy with lower dose, this guy with higher dose, or this guy with blue light, or this guy with green light, or the one activate drug, the other one is not activating the drug. You can actually do them simultaneously in the same field of view. Before you have to do it one at a time. Sometimes you need to monitor it 24 hours. You can imagine the throughput is very low. Now it's very high because you can do them all together simultaneously for all the conditions. So there are many applications. For example, you can look into this wrap. I'm not sure if you're familiar with that or not. Basically, you photo bleach something and see how the molecule refill that area, so to see the dynamics of the protein. So usually people photo bleach here and then move to here. Let's say you have multiple positions. So once you photo bleach the last one, you realize the first one is already recovered. So sometimes it happens because the recovery can be fast. In our case, we're not controlling the gobble mirrors, we're controlling the lasers. The image is always on, it's constantly scanning. It's just once the laser goes there, it will turn it on or off. So basically, you will not hamper the imaging at all. So you can capture any single moment through this entire protein bleaching and recovery process. So that's the one advantage. And we can also just outline a cell. Let's say I want to treat the mitochondria in this cell, and then I just outline it and then use the mitochondrial signal to damage the cell with blue light. Generating ROS, you can see the membrane potential is actually damaged, so you can see the dye leaking and oxidation of this GFP molecules. This is actually more obvious. If you look at, I outline this cell, I turn on the drug in it, so this cell is microtubule polymerization is inhibited, and other cells are still active. So this is you can do with any cells in the whole population, or even do a long-term study. I can monitor this whole field of view for like even 72 hours if you want, to see how the cell, after you perturb the nucleus, at what dose they will trigger the apoptosis, at what dose it will be able to revert it, which is at what dose is safe and what dose is really toxic for cells. Or control the cell division, for example. This is a dividing cell, okay? Nucleus, well, you know the DNA is a lining, and these are the microtubule spindles. By tracing this, you can find the centrosomes. If you treat the centrosome with the blue light and the green light, you can delay the whole mitotic process. And you can see actually how this blue light treatment messed up the spindle of the microtubule during the mitotic phase. And this actually will trigger the cell to develop into a multi-nucleus configuration. But you can also do it for trigger mitochondria. You can also treat mitochondria in other organelles and see how that can control the division of a cell, and then how that, if the daughter cells are affected or not for both of the daughter cells. So those, I don't have time to show these data, to show those data, but I do want to go back and to show you one thing is we talked about this earlier, right? So we want to inhibit the microtubule polymer, we want to inhibit the mitochondrial function in the protrusions and see how that affects the microtubule polymerizations. So what you see is if you damage part of the mitochondria here, you will actually, so this microtubule polymerization is gone for that area. Usually this dynamic is very fast. It's recovered in, if you don't have mitochondria there, it recovers in like 10 seconds because the microtubule dynamics, tubulin dynamics is very fast. So, but it takes about 10 to even 30 minutes actually for this process to recover. And there's another one that's even more obvious. It takes about 30 minutes. If you damage the mitochondria here, the microtubule polymerization will be gone until there are new mitochondria migrate into this place, and then you can start the microtubule polymerization start to recover. So this is the only way you can do it, because you cannot use any other techniques to really only perturb the microtubule function only here without affecting other places. So it helps us to answer a lot of important questions. So we haven't published this one yet, but it's in preparation. So we did a lot of calcium imaging, like in live animals. Like this is a living, this is a zebrafish embryo. So I probably want to cut it short. So this is basically what we're trying to do is we try to see if we uncage some small molecules here, how that triggers the calcium waves to mimic the wounding effect. So if you uncage the ATP here, it will trigger the calcium wave, and the animals sense it, and they start a repairing process. So usually, this is a GCAM modified fish, by the way. So usually people do it in the confocal microscope. You uncage the ATP somewhere here, and then later comes back to see how this calcium wave is propagating. But that doesn't work for them. The reason is it's unpredictable. You don't know how long it takes for how much ATP is needed here for this to trigger this process. So sometimes they do this triggering and then come back, the wave is already propagated, or the wave is not even triggered. So they need a way to simultaneously monitoring this whole process while engaging the ATP. That's perfectly for us. That's why they collaborate with us. And they also wanted to stop this treatment right away after you trigger the calcium waves. And that's better mimicking the winning effect. So, and you can see that's why this RPOC technology is perfect for that application. We can kill the cells you don't want precisely. Let's say you have two cells, cancer cells and healthy cells. And cancer cells are labeled with some surface marker, or you can use some dyes to label it. And just scan it with blue light for like 2 minutes. You kill all the cancer cells. They're all round up and membrane blabbing, and healthy cells are not even affected because you don't even shine laser to it. So physically they're separated. So I hope that this can improve the photodynamic therapy efficacy and the precision for it. So we're still exploring this. I probably don't have time to cover details of this one. But I do want to mention one thing is here, we can also use a femtosecond laser as the action laser. Basically, femtosecond laser can also be used to treat something. Before you only saw the CW laser, different colors. But the femtosecond laser work in a different way. They generate a multiphoton process. You can generate low density plasma. So basically, they generate a higher energy, the molecules with higher reactivity, but they're more confined in the focus because it's a multi-photon process. So it's more three-dimensionally defined. So I'll just skip this part. I work with a little bit with Jixin Cheng from Boston University, and he used our device to do cell sorting. So basically, he's sorting bacteria that's moving in the solution or moving in the dish. So you can't take an image and come back to sort it because this bacteria is gone next to frame. So he has to use this real-time feedback system to sort bacteria. That's the first comparator circuit we give to them, and they get this one done. So we have a startup company called Photokinesis, as Susan introduced. So we have the product of this comparator circuit and the software system. So if you have any laser scanning system that you are interested in converting into an opto control system, you only need these two components. Just add it into your system. That's it. So you can use all kinds of action laser to do whatever you want. So it depends on your need. And if you don't have any expertise in lasers and spectroscopy, we're also almost finishing up this last platform, which is we call Sentry 1. So this system is more advanced. So all the lasers now are fiber laser coupled. You just need to screw it in. There's no alignment at all. So basically just put the laser in and then screw it. There's a fiber coupler there. And then the feedback is even faster than 20 nanoseconds because we completely get rid of AOM now. So we don't need an acoustic optical modulator, which you have to generate the acoustic wave, have to propagate. So this is directly controlling the laser. So the response time for the laser is 2 nanoseconds. So if you're adding other things, it's about 20 nanoseconds. So it's very fast. So also, this is still a confocal microscope. You can use it as a confocal fluorescent microscope. And I think it's cheaper than whatever is on the market. So anyway, so I'm not going to go through this page again. So, but I want to say one word that this part is more like watching a movie because you're basically looking at what's going on in the sample. So, it's good. I like it. And you can use all fancy ways of doing it, super resolution, chemical imaging. But the experience of this one is very different. This is more like playing a game, right? So you're involving in the process of this chemical reactions in the cells. You can actually control where chemical reaction is going to happen or be inhibited. So then you see how the cell responds to that process. So I hope more people will be interested in joining this, doing more control of the chemical reactions, not just simply watching them. So that's we're hoping that we're trying to promote this technology to a broader audience. All right, so do I still have time? Yeah, anyway, I will just go through this acknowledgement. So I want to acknowledge my group members. Some of them are already graduated, and those highlighted ones contribute a lot to this, the results you have seen. And our collaborators, and especially the AMI facility folks who help us to design a lot of electronics, software, and hardware. And the funding agencies, especially the NHMS, who is the major patron. And we have the company called Photokinesis. And thanks, Lucian, again for inviting me. And thank all of you for your time. And I'm happy to answer any questions. It's not too fast. Yes?
So that device you showed at the very end where you were selecting bacterial cells, how do you actually isolate physically the cells?
Oh, that's a good question. Although I didn't do that experiment. It's actually completely done by Jixin's lab, and I'm only offering them the device. But as far as I know, they use a laser. They have a polymer or like some kind of substrate. When you shine a laser, you can eject it. So basically, you put a green light on it. This thing will fall off. So the bacteria on it will just fall with it. So they have a dish underneath to capture it. And then after that, they basically just sort the interest and grow back into colonies and see how the properties of this bacteria. And they do all this. I think they did some like a genetic sequencing or something to see the species of this. I mean, the specific genome changes of this bacteria. So that's as far as I know. But I didn't involve it too much on that project. Yeah. Thanks.
Curious about generally, based on your experiment, are there any current bottlenecks of the parameters of the microscope you're using now that prohibit you from identifying problems, questions in biology, say maybe lack of resolution or the penetration depth?
That's a great question. So we actually currently we use the imaging system all kind of a confocal based or diffraction limit based, I would say. It's not super resolution imaging. And even if you do super resolution, it doesn't work at this point, it doesn't work for the control. Because you can generate very good images, high resolution images, but your action laser still diffraction limited, right? It still works that way. When we were thinking of this, we can use a donut ring or something, inactivate something outside and activate inside. Those are just concepts now, but we haven't really achieved better than the diffraction limit precision. But I would say, in terms of imaging, it reaches the resolution of the diffraction limit. And in terms of optical control, it also reaches the diffraction limit. So that's the maximum we reached. And we actually adopted all these advanced chemical imaging approaches. Whatever that chemical imaging approaches can reach, then we usually can reach the same level. But we can't really get the below diffraction limit. And in terms of scattering, if you go deeper inside, that's the general problem for almost all the optical techniques, right? So you basically scattering will make your resolution worse. So if you go deeper, so you have to expect the precision or the resolution gonna get worse for this one too. So it's basically the same as other optical imaging tools. And if you want to do it better, you can use a near IR or longer wavelengths or multiphoton like those. It still applies here. Yeah.
I have another question. During your path of commercialization, do you find there's something that actually, like research field cares more, but the industry does not care, and the opposite way?
Oh, great question. That's very general, right? So the research people care about papers, I think. Company people care about simplicity, I would say. Because the more complicated you get, the less application. The more problem is going to get, you know. I talk to companies, I agree with them. So usually, whatever lasts longer, the longest is actually the ones that is the most simple. So that's why we're moving from SRS back to this simple confocal microscope. I'm a student, that's a downside for research groups. I agree, because my students, once they start to work on the fluorescence, they don't want to work on SRS anymore. So I'm the only 2 people who can work on SRS right now. Because it's a tendency of people like to do something relatively simpler and robust, so they don't have to spend half a day to tune something. So that's the expectation of industry, I think. They want something to solve their specific problem. They don't care how complicated it is, or if it's complicated, you can solve their problem, it's still fine. But sometimes they wanted to be able to adopt it. So they wanted to use it in their labs to do it. If it's too complicated, you have to hire a person there to just work on that thing. It's probably not going to work. So You might hear different opinions. That's just my personal opinion. Yeah.
Are there any restrictions on the wavelengths you can use for the action laser showing transfer?
Great question. Actually, no. As long as you can deliver it to the sample. We actually tried IR. You can hit the specific organelle. You can basically damage it that way. And you can you can deliver the femtosecond laser to it. We're actually doing another one where I didn't show here. You can actually deliver different wavelengths with a continuous wave or with a tunable laser so that you can generate spectrum. So once you generate spectrum, you can see is this, what is the mechanism of that interaction? For example, I said 405 interact with FAD and generate our ROS. Is this really the case? So if you take the spectrum of it, it matches the FAD's absorption spectrum, that means it's right? So if you have a higher absorption at 420, that means it is mediated through FAD. So otherwise, it may be NADH or something. Those can be done with tuning the color of the laser. But that's tedious, Each one, probably you have to monitor for some time. And if you do the wavelengths changing, it's probably going to take an even longer time. But it's doable, yeah. Thanks.
Okay, one more question.
Yeah.
I guess in your experience, which one is more phototoxic? Is it confocal? Is it SRS? Is it multiphoton fluorescence?
Great question. Visible light is way more toxic than IR. So I usually use IR. I show that you can use 24 milliwatts of IR to image sample. It doesn't matter, right? For visible light, we use 800 microwatt to treat the cells, basically to kill the cells. As you can see, the live cell can only sustain less than 1 milliwatt of visible light. Because of this, 4 or 5. Maybe 532 can be toxicity is less, but visible beam is very toxic because the intrinsic fluorophores work as photosensitizers. So basically FAD, NADH, or maybe some cytochrome C or something, maybe. So they basically absorb the light and then give that electron to oxygen and then convert it to ROS. So that's why, because IR doesn't do it, right? IR doesn't much less efficiency. So if you use IR, so it's more safe. You can go up to 20 milliwatt or something. But I haven't tried the long-term imaging. But visible light, you have to keep it. Usually we use like a 20 microwatt or something for visible light in order to keep the cell happy for long term.
Then how do you keep the cells happy when you are using confocal to scan the active area?
Just use the power very low. So we usually use less than maybe 100 microwatt or something. So it's still perturbs it, but you can see actually it goes up. I actually want to show you one thing. I'm not sure if you noticed that or not. So this one right here. So you can see this thing goes darker and then goes brighter, right? That's because your image is too frequent here, but it's recover. It can recover. So the confocal fluorescence is toxic, but it's less toxic if you only work with fluorescent proteins, because fluorescent proteins are beta sheet wrapped fluorophore inside. The fluorophore photosensitization is not really that efficient compared to organic dyes. If you put the metal trigger here, the cell will die right away. So it's very, very toxic. So.
Okay,
Yeah.
let's thank Dr. Zhang again for giving us this great lecture.
Thank you very much.